All right, so today's lecture is about genome editing. And you'll hear the molecular biologists, biotechnologists, and economists. Everybody is very excited about this technology. It's going to be as transformational as PCR was to molecular biology just a few decades ago. So we're going to talk about some of the different techniques that are used for genome editing, and then we're going to push our focus to mostly this technique called CRISPR, which uses a guide RNA to make genome editing, which is very versatile and has been basically taken over a lot of the other genome editing techniques. All right, so what do I mean by genome editing? This is what I mean. It's the use of programmable nucleases that enable targeted genetic modifications in cultured cells as well as in whole animals and plants. What's very exciting about this, in addition to a lot of other things, is that you can use these techniques not only in model organisms. So it really gives us a way to study other organisms using molecular biology as well, as long as you can inject or transfer DNA or protein or RNA into the germ cells or the eggs or the embryo of any organism, then you can modify their genome. Okay? So this includes mosquitoes, silkworms, pig, cow, non-human primates, as well as humans. All right. So what are the different types of genome editing techniques? These are classified into three in this lecture. We're going to talk about how you can use zinc finger nucleases to edit the genome, use transcription activator like effector nucleases, TALENs, and then RNA-guided engineered nucleases derived from the bacterial, clustered, regularly interspaced, short palindromic repeat. So it's shortened as CRISPR. It's easy to remember. And Cas9. It's a type of enzyme that's associated with the CRISPR. All right, so what are the type of things can you do if you edit the genome? The simplest thing you can do is cause mutations, right? We know that double-strand breaks can cause mutations through non-homologous end repair or homologous recombination. So if you have the ability to target a double-strand break to a genomic site, this gives you enormous power to make mutations. So if it goes through the non-homologous end joining pathway, you can introduce deletions or small insertions, and these are variable length. You can also use a donor template and use the homologous recombination system inside the cells to be able to copy part of the regions of whatever you want to insert into the genome, right? And both of these techniques you covered with Professor Siegel and with me, so I'm not going to go through them. So what can you use this? You could use this for a lot of different things. You can introduce gene mutations, or you can do knockouts. You could do replacements. And this kind of genetics is a little bit different from classical genetics, right? So what would you call this? Is this forward genetics or reverse genetics? Reverse, right? Because we are going from the knowledge of the sequence back towards a phenotype. If you don't know the sequence, you can't really target a specific site in the genome. So we need to know the sequence, and I'll tell you why in a minute. All right. So this is the first group of proteins system that we can use for genome editing, or you could call it gene editing. Genome editing is more precise, I would say. So what this takes advantage of is it takes advantage of our knowledge of proteins that bind to specific DNA sequences, right? And these proteins are basically the transcription factors, sequence-specific transcription factors, and they have specific protein domains that can bind and recognize certain sequences in the DNA, right? And one of these protein domains that we learned in transcription factor lecture was that a little motif of DNA, a little protein structure, right, called a zinc finger. And that zinc finger actually sat on the DNA and recognized the DNA sequence. So in this case, what the researchers have done is they took zinc fingers that they know each recognized 3 base pairs, okay? So they stitched several zinc finger domains to each other, and each of these zinc finger domains are going to recognize 3 base pairs each, right? And the way that you stitch them and fuse them together, you can make a set of these zinc finger proteins to recognize a specific sequence. Is this clear to everyone? So you basically engineer the protein such that you fuse different zinc fingers that each recognize a 3 base pair sequence. And depending on how you combine these zinc fingers, you can change the sequence that it is going to target. Now, that zinc finger protein is fused to a nuclease domain of a protein called FOC1. So this protein nuclease is interesting because it's an endonuclease, but only works when 2 FOC1 domains are dimerized. Okay, so it's going to work when it dimerizes together. So what you do is you also then create a protein, fused protein, where you target the sequence on the other side of the DNA through the recognition of each of these 3 base pair elements with an individual zinc finger protein that are fused together. So the spacer between these 2 recognition sequences should be about 5 to 7 base pair, and that allows the FOC1 nuclease to dimerize and cut right here. Okay? So 2 ZFN FOC1 monomers form an active nuclease and target sequences about 5 to 7 base pair apart. And here is the approximate crystal structure, filled in crystal structure of this with the enzyme and the zinc finger nucleases, zinc fingers, etc. Any questions about this? So you can engineer the protein so that you can then put it, transfect into the cells, and if you do it right, 2 of them are going to get together and cut it. Okay? All right. How can you use these zinc finger nucleases? Here's a proof of principle experiment from quite a while ago. I guess like 6-7 years ago. I can't remember. So what they've done here is they said, okay, we know that there are CD4 plus T cells that can be made resistant to this HIV virus. Okay, and they know the molecular mechanism behind this resistance. It is due to a 32 amino acid deletion in the HIV receptor CCR5. Okay, so if a CD4 cell contains this mutant allele of CCR5, then it's resistant to HIV. So they asked, can we make these resistant CD4 T cells in vitro and then inject it into the mouse and see if the mouse now becomes HIV resistant, right? And so what they did is they used the zinc finger nucleases to make this precise deletion, and then after they made these precise deletion, they put it into a model. So this is a humanized mouse model of HIV infection, and the mice that had the zinc finger nuclease modified CD4 T cells then had lower viral loads, suggesting that it now has more resistance to HIV infection, right? So a system like this can be then applied instead of mouse to humans to combat the HIV disease. All right. So you can use this for what we call gene therapy, right? You change the gene of the organism as a therapy method to correct for that disease. So that's zinc finger nucleases. So it uses the transcription factor binding domain. And we saw that the transcription factor DNA binding domains can be of different kinds, right? And one of these kinds is here. And in this case, instead of each protein domain, so shown in like little colored oval shape here, instead of each recognizing 3 base pairs, these protein motifs recognize 1 base pair at a time. So you can design these proteins so that each of these each of these protein domains are going to recognize 1 nucleotide sequence, and the combination, you can combine them in such a way that you can target a specific sequence. And again, just like the zinc finger nucleases, these are fused to the FOC1 endonuclease. When it dimerizes, given a certain spacer, in this case it's a little bit larger, you can cut the DNA right here. So these are called transcription activator-like effector nucleases. So in short, they're called TALENs. So the difference between zinc finger nucleases is that they use a different domain to bind the DNA and recognize the DNA, and that occurs at 1 nucleotide at a time. Okay, so the same principle. So each of these domains are actually 33 to 35 amino acid repeats, and each of them recognize a single base pair in a major group. Any question about this? All right. So what you're doing is you're taking advantage of the fact that you know something about protein structures that can recognize specific sequences, and you engineer it in such a way that you can make them bind to a certain sequence, right? All right. CRISPR is actually completely different. So they're different CRISPR systems, and I'm going to go through only one of them that's the most commonly used one. They're called the RNA-guided engineered nucleases. So CRISPR-Cas9 system is one of them. And it includes a protein, an enzyme called Cas9, shown with this very light blue here. It probably shows better in your printouts or your computers. So this Cas9 protein associates with the RNA. So it associates with RNA. So this is the RNA here. And this is a typo. These T should be used, okay? So it was a typo from the actual publication itself. Anyway, so this is the RNA. That is called the recognition RNA. Okay. So this is the RNA. And there's also a second RNA that is more structural. It has certain stem loop structures, and this is called the tracer RNA. So the Cas9 protein forms a complex with these 2 RNAs. And when it forms a complex, it can go into the genome, and this is the double-stranded DNA here, shown here, and it looks for homology, so it looks for complementary sequences that can complement the guide sequence, and that guide sequence is 20 base pair long. Now, when it finds this guide sequence, it can actually bind to this DNA by forming this complementary annealing, so that you're going to basically have a bubble here, right? And instead of this DNA sequence being in complex with the other strand of DNA, it's going to get into a complex with the RNA that is associated with Cas9. And what it does is it cuts, the Cas9 enzyme cuts both strands one at a time right upstream of this sequence that is called the PAM sequence. That is the NGG, so it can be any nucleotide that is followed by 2 Gs. All right, so this 20 base pair region here is called the protospacer, and I'm going to tell you why that is, because it's from its bacterial origin. And the PAM sequence is named because it's protospacer adjacent motif. Okay, so now this also works if you actually fuse the RNA molecule that serves as the guide sequence directly to the tracer RNA and form a RNA molecule that's called the single guide RNA. Okay? So the Cas9 now can make a complex with the single RNA molecule, the single chain of RNA molecule, and bind and cut the DNA very similarly. How does the structure look like? It looks amazing. What it looks like is that it is a protein that can surround the whole DNA double helix here it can sit on it, and then it cuts the DNA right upstream of that PAM site. So 3 nucleotides upstream of it. It's a clean double-stranded break. All right. First of all, can you guys tell me what's the difference between how the recognition occurs for the CRISPR system versus the ZFN and TALEN systems? There is a drastic difference how the DNA recognition occurs. How does the ZFNs and TALENs bind to DNA? What's binding and recognizing the sequence? Protein, right? The protein domains are binding and recognizing the sequence in TALENs and ZFNs. What's binding and recognizing the sequence in CRISPR? RNA. So that's a huge difference in which how the recognition occurs, right? All right, here's another question. So the recognition in CRISPR is occurring through recognizing a 20 base pair protospacer, right? So that's what the guide RNA is recognizing and binding complementary to. We call an effect called off-target effect. That means sometimes protein, instead of recognizing exactly its own sequence, instead of cutting the region where you're sending your Cas9 to, right? So you are targeting a specific genomic site. It can sometimes cut by accident other sites in the genome. Okay, so that's called the off-target effect. Why or how can this happen? You guys learned PCR. You guys learned how PCR primers work. Works very similar, right? Short oligonucleotides binding to the DNA in the genome. Tell me how off-target effects can occur with CRISPR editing.
There's another piece of DNA that matches the strand of RNA, and it'll cut there too.
If there's a piece of DNA that exactly matches the RNA sequence that you're using, but you're just not aware of that being in the genome, it can cut, right? But you're pretty smart. Like before you actually design your guide RNA, you should do a blast and search that, okay, this guide RNA is present only at the site that I'm actually sending this thing to. But sometimes, even if the sequence is exactly not the same somewhere else, if there is only one or nucleotide differences, right, the guide RNA can still complementarily bind, perhaps not as efficiently because it's not going to be perfect complementation, right? But it's going to be maybe 1 or 2 nucleotides that's going to be not binding. But if it binds just enough, that Cas9 will be able to cut there as well. So that kind of off-target can occur. Same thing with PCR primers. Even if it's not perfectly complementary, if there's 1 or 2 nucleotides that is not complementary, it might still bind somewhere else in the genome, and you're going to get some background bands that is not specific. Okay? So when you're designing your PCR primers, once you're in labs, just make sure that your primers is not blasting somewhere else in the genome. And not only it's not blasting, that it's at least a few nucleotides different from other regions in the genome, right? Okay. So this CRISPR has been discovered in bacterial immune systems by a number of groups of scientists, but 2 of them actually stands out from this crowd because they actually recognize the applicability of this technology. Okay? And these people who worked out the molecular biology of the Cas9 enzyme and talked about the application to a lot of molecular biology are these scientists, Jennifer Doudna and Emmanuel Charpentier, and they've been somewhat of a celebrity in the scientific community. Jennifer Doudna was here last year during our genome symposium, and it was very exciting. All right. So what were they studying? They were not studying to cure cancer or anything like that. What they were studying was they were studying bacterial immune system, and they recognized that there was a very particular and interesting mechanism that is being going on here. Okay, so what they realized is the bacteria has an immune response to viral DNA infection. So once a virus infects the bacteria and it releases its viral DNA in its in the cell, what happened is that there is a protein complex, this Cas9, and the RNA complex actually cleaved the viral DNA, and it took a small DNA fragment that is called the spacer. That's the 20 base pair that I'm talking about. It took that small DNA fragment and inserted it in its own genome, okay, at a site where you see various repeats of these type of 20 base pair sequences. These are different sequences, but there are 20 base pair of each of them, and they are actually surrounded by these repeat sequences that are same. So you have 20 base pair, a small repeat, 20 base pair, a small repeat, a 20 base pair, etc., etc. And these 20 base pairs 20 base pair spacers, interestingly, correspond to the various viruses that this bacteria is infected. Okay, so it's kind of a history of that bacteria's infection. And this CRISPR locus here, there are different types of CRISPR. This is a type 2 CRISPR locus, also contains the gene that encodes for the tracer RNA. It encodes for the Cas9 enzyme and several other elements that I don't want to get into right now. All right, so I have some notes here. Okay, so this system basically provides adaptive immunity because it's adaptive, because once you get infected, then you can actually respond next time to invading phages or plasmids. So the bacteria capture small DNA fragments from the foreign DNA and insert into their own genome. All right, so what happens when infection happens the next time? So the virus infects the cell, but now the cell, what it does is that these CRISPR repeats are transcribed, and then they're processed and loaded into the Cas9 enzyme right here, along with the tracer RNA. So remember, the tracer RNA and the guide RNA forms a complex, and once these complexes are formed right here, and the mature CRISPR RNA, tracer RNA, and Cas9 complex is formed, it can target the viral DNA and cut it. Okay? And the virus will no longer be able to make its own proteins and infect the cell. Is that clear to everyone? Any questions about this? All right, then I have a question for you. The same guide sequence that is in the Cas9 is also here. So why it's not cutting itself? The reason why it's not cutting itself is because the context in which the sequence occurs is not the same in the bacterial genome. So while in the target DNA, this CRISPR RNA has the repeat sequence that is going to be not complementary to the target DNA, that 20 base pair spacer is going to be perfectly complementary to the target, and now you're going to have a hairpin structure, and this is going to form the complex along with the tracer RNA with the Cas9, and the Cas9 will have the correct conform' to be able to cut the target DNA. However, in the CRISPR DNA itself, so in the bacterial DNA itself, this little 5' extension of the CRISPR RNA actually is also complementary to the repeat sequence, and the Cas9 CRISPR RNA tracer RNA complex forms a structure that is incapable of cutting the DNA here, right? So the bacteria, while it has the spacer sequence, protect itself from cutting to make sure to have that little flap that is going to prevent the Cas9 enzyme to cut itself. All right. This is very analogous to another mechanism by which bacteria can fight plasmids and invading viral DNA, and those are the restriction enzymes, right? So you've learned about those. So you learned about, for example, EcoR1, one of the first restriction enzymes to be discovered. It cuts the DNA, right, of the invading plasmid or the virus, but it doesn't cut the bacterial DNA itself. Bacteria protect itself from the potential cuts the restriction enzymes can make. How do they do that? Did you guys study the study question of methylation? Thank you, Alex, right? So what they do is they methylate their own DNA, so the EcoR1 cannot cut the bacterial DNA, right? So if you have a response to virus, you need to also protect your own genome. All right. So what can you do with Cas9? Did I make a big deal out of Jennifer Doudna and Emmanuelle Charpentier and showed them in their best outfit? Because they recognize that you can use this system for a wide variety of things in molecular biology. You can use it for reverse genetics, and we talked about that. You can use it for transcription regulation of individual genes. You can turn on or off genes depending on where you send that Cas9 and how you engineer it. You can visualize genomic loci live under microscope. So instead of using immunofluorescence techniques, which requires cells to be fixed, you can actually watch individual genomic loci in the cell. And there are also some ethical concerns that I'm not going to dwell too much about. All right. So the genetic, you can do reverse genetics, which is that you can you can engineer a guide RNA, right? You can engineer a single guide RNA that will have the 20 base pair spacer that is complementary to wherever region of the genome that you want to send this enzyme to, and introduce a cut over there. And so it introduces a cut, and you can let it just repair itself and hope for some mutations or screen for some mutations, or you can make sure that you have a DNA template that can be used for homologous recombination, in which you can introduce a selection marker, another gene next to a selection marker, etc. Very similar to what you guys learned in mouse genetics, but you just use Cas9 as the tool to introduce the double strand break. All right. How can we use this? And do we use this in our own research? Yes. We've been using this in the past 3 years or so, and it made our lives very easy and very happy because we can do things so much faster than we used to do before. What do we do now? So in the past few lectures, I've been talking about this dosage compensation complex, which contains these proteins called SDC proteins that bind to the genome. And here is a ChIP-seq profile of one of these proteins, where you can see that this protein is binding to these two sites in this part of the X chromosome. And we learned that these two sites contain these DNA sequence motifs called about a 10-12 base pair motif. That is, the core of it is a GC, GC, A, G, G, G. So my lab said, okay, is this motif actually important for that protein to bind there? And what you can do is then you can introduce a mutation. So what we did is these two binding sites were actually an inverted repeat. So we had a repeat in the genome and another repeat that was inverted. And we sent guide RNAs right next to the motif. And we knew that the Cas9 enzyme was going to cut somewhere around the motif, and we said, okay, let the non-homologous end joining pathway repair this and hope for some mutation. Then we had to screen the resulting C. elegans animals and look for the mutation. And we were hoping for small deletions. And how do you see small deletions? What you can do is you can design 2 PCR primers. So in this repeat, we designed this primer and then that primer, right? That's going to amplify this short DNA fragment. So in wild type, we should see about 685 base pair fragment. If the mutation here that's introduced by Cas9 causes a deletion, then I would expect a smaller fragment size, right? So then if I take the progeny of C. elegans that we actually edited, and then look for the size of the DNA fragments that are coming from the progeny. So here's the wild type. So that's our positive control. It should be 685 base pair. And then these are the progeny that we screened. And you can see that these first 5 have exactly the same size as the wild type. So even if it contains a mutation, it's not a deletion, right? And then we were lucky enough to get one that looked slightly smaller. And when we went back and did a Sanger sequencing to see what kind of mutation we introduced, it was about a 20 base pair deletion that picked out a pretty good chunk of the motif. Okay. Then what we did is we made sure that the other repeat containing the motif was also gone. And then what we did is we did a ChIP-seq experiment. So if the motif at those 2 repeats, if those motifs were actually important, when you deleted them, you should see that the protein should not be able to bind to this site anymore, right? And here's the wild-type binding pattern by ChIP-seq, and you look at the mutant binding pattern by ChIP-seq, and you can see that this binding here is mostly gone. What you see here is just the background from the ChIP-seq experiment. This is the background data, how background data would look like, right? So this is how you can use that kind of technology in our research and answer questions. In our case, it was, is this motif actually important for the recruitment of the dosage compensation complex? All right. Next, people who work on crystal structure of the Cas9 enzyme mapped the enzymatic sites, the catalytic sites of this enzyme, and made 2 very specific mutations that completely eliminated its ability to cut the DNA, but it did not eliminate its ability to bind to DNA. Okay? So now what you created is a protein where you can target things, but not necessarily introduce mutations, right? This enzyme is called dCas9, stands for dead Cas9. It's enzymatically dead, but it is able to bind to DNA. And the structure looks something like this. What you have here is you have the single guide RNA that's shown in red here, and here is the structural portion of the RNA. The single guide RNA is making the complementary interactions with the DNA that's shown in yellow and blue here. And the 2 amino acids that were changed were histidine and what does D stand for? I forget. Aspartic acid, I think. I don't know. Changed to alanine. Okay, so these single amino acid mutations basically made the catalytic domain completely inactive. So now the enzyme with the single guide RNA can bind to DNA, but does not cut it. So what do we use this for? You can use this for a lot of different things. Remember, transcription factors regulate transcription through 2 distinct domains, right? In one, you have the DNA binding domain that targets a specific DNA sequence, and there was a separable activation or a repression domain that act to regulate the transcription. Now, if you can take a transcription factor and only take the activation domain and now fuse that to dCas9, what you've done is you target the activation domain to any region of the genome that you want, and then you can modulate transcription, right? So it's kind of what is drawn here. What you have here is you have the DNA, you have a target gene that you want to repress or activate, right? And what you can do is you can engineer and make a single guide RNA that has that spacer sequence that is complementary to upstream or wherever you want to target this dCas9 of the target gene. So if you want to activate or repress, you would probably want to target the promoter region of the gene, right? So you make an sgRNA that's complementary to the promoter region. Now this dCas9 will be protein will be fused, so you're going to express a gene that is going to encode for dCas9 fused to some kind of effector protein domain. And that effector protein domain, if it's a repressor domain, it might repress transcription, and if it's an activator domain, it might activate transcription. So what do you need to do to do this in cells? What you can do is you can make a plasmid that contains a gene that encodes this dCas9 fused to effector. You also make another plasmid that encodes a gene that is, when transcribed, makes this single guide RNA. And this green region is the region that's the 20 base pair that's going to be complementary to your target site. And then you can take these two plasmids and transfect into the cells, and then the cells will express these, and then you're going to see if you can activate or repress transcription. So now let's look at some real data. Okay, so this is from a study that actually showed this as a proof of principle. So what we have here is that 3 constructs. What they've done is they created this sgRNA construct that is going to target a specific gene. In this case, a reporter gene. Okay? And then what they do here is they created another construct that's going to encode for the dCas9 protein. They are putting a nuclear localization signal. Okay, so this is nuclear localization signal is a short stretch of amino acid that makes sure that once the protein is translated in the cytoplasm, it is transported into the nucleus. Not every protein in the cell is nuclear, right? Like histones are nuclear, RNA-pol2 are nuclear, but other proteins, since I work on the nucleus, I can only think of nuclear proteins coming up in my head. But other proteins that are strictly cytoplasmic do not contain this nuclear localization signal. So they want to send the dCas9 into the nucleus. They put this nuclear localization signal. And then they fuse this followed by a blue fluorescent protein. So they can see the protein inside the nucleus, right? And make sure that it's where it's supposed to go to. And then they fuse to a protein domain called the CRAB domain. It's a repressor domain from a transcription factor called CRAB. So as a control, so you want to control your experiment. You want to know, if I fuse this CRAB domain, do I then repress the transcription of my target gene? What you can do is you can make a control where you make exactly the same construct, but you don't add the CRAB repressor domain. Okay? So now what you can do is you can compare the effect of this to this, right? All right. So next what they did is they also cloned a reporter gene. So instead of targeting it to a specific gene in the genome, they want to show that this can work as a proof of principle. So what they did is they made a plasmid that contains a reporter gene. And in this case, you have the promoter here. It's a basal promoter. And then you have the 5' UTR. You have the GFP, a version of the GFP protein. And when you see the GFP signal, you know that this gene is expressed. And when you don't see the signal, you know that this gene is not expressed. And this promoter, by the way, is a very active promoter. So by itself, it's going to support a lot of GFP expression. Okay? All right. So next, what do you do? What you do is you express your sgRNA with one of these proteins. So that is going to target the promoter. So they made the sgRNA against the promoter site of the reporter gene. Then they co-transfected with it either dCas9 blue fluorescent protein or dCas9 blue fluorescent protein followed by CRAB. And then they measure the GFP. All right, here's the actual data itself. In gray is shown the no reporter. So there is no reporter cloned put in these transfected cells, so you should not see any GFP signal at all, right? So that's going to show you baseline, no expression whatsoever. So what we have is on the y-axis counts. That's counting how much GFP is expressed. On in the x-axis you have that's counting how many cells are expressing the GFP, and then on the x-axis is you have the fluorescence. So the higher the fluorescence, the more GFP is expressed. Does that make sense? And in the gray, what you can see is that there is very, very little fluorescence, right? All of these cells are not expressing the GFP. Does that make sense? All right. So next what they say is, okay, let's see, if we put our reporter, now this time we're actually putting the GFP gene with the reporter, then we also put in dCas9 that is fused to the CRAB domain, okay? That's fused to the repressor. But you don't put, you don't put any sgRNA, right? And if you don't put any sgRNA, what's going to happen? The Cas9 sgRNA is not going to bind to the promoter, and you expect that the expression of the GFP should not be affected. It should be expressed pretty highly, right? And that's what you see here. So what you see is that the expression of GFP has gone much, much higher compared to the control, because the GFP is expressed. Without the sgRNA, you can't target the Cas9. All right? Now, let's put the same setup, but this time, in addition to the dCas9 CRAB, you actually add the sgRNA. Now, you targeted the complex to the promoter of the reporter gene. And what happens is that from being expressed, it goes down to the background level, right? So there is no GFP expression. What this tells you is that the dCas9 fused to the CRAB was capable of completely silencing the reporter gene, in this case the GFP, correct? All right. So what's happening in the purple one? In the purple one, what they did is they did something curious. They said, okay, I'm testing the effect of the repressor, right? And they also made the construct without the repressor domain that's fused. So you have the dCas9, and they injected it with the reporter and with the sgRNA. So the dCas9 is binding to the promoter, but is not fused to the CRAB repressor domain. And you look at the amount of GFP expression, and then you see that it's a little bit lower than how it would be expressed if there was no repression whatsoever. What does this result tell you? Cas9 by itself was capable of repression to some extent, right? Because it went from here to here. Does that make sense? Any questions about this? It's really important, guys, that you can read a data and really synthesize in your head and know what the data is telling you, right? Okay, so I just told you that Cas9 by itself, even without fusing to the repressor domain, if it binds to the promoter of this reporter GFP gene, it repressed it to some extent. Not completely, but it caused some repression. Knowing how eukaryotic transcription factors work, knowing how lac repressors repress transcription, right? Can you guys tell me, given hypothesis, by which this binding at the promoter might be repressing transcription without the repressor domain? It could be sitting on an activator site, right? It could be sitting on a site where the general transcription factor, initiation factors need to bind and start transcription. So it might physically impede the initiation complex to start forming. And that's exactly how lac repressors work, right? They sit into where the sigma factor is supposed to bind, and they repress transcription. So even without the CRAB repressor domain, it's capable of repressing a little bit, but with the action of the repressor domain, which might include recruiting chromatin modifiers that are going to deacetylate the nucleosomes around here, make sure that the nucleosomes are not removed, you can further repress even more to the absolute background levels, right? Does that make sense? All right. If that makes sense, then we should go through this experiment even easier. In this case, what they're doing is instead of looking at repression, they're going to be looking at activation. Okay? So instead of fusing a repressor domain, they're going to fuse an activator domain. So in this case, what they're doing is they have cloned a reporter gene with the GFP, but this time they're using a weak promoter, right? Because we're going to measure activation, you don't want your baseline level to be lots of transcription. You want to keep it very, very low, right? So you have a weak promoter. And upstream of that promoter, they cloned a whole bunch of sgRNA target sites. Okay, so these are artificial sites, and they cloned many of them because they already knew that it wasn't working so well. Anyway, so they cloned a whole bunch of them so they can target multiple Cas9 proteins. And at the same time, they made the plasmid that's going to encode for the single guide RNA that is going to target the dCas9 over here. And then here, what they did is they made the plasmid encoding for dCas9, fused to the nuclear localization signal, fused to a transcription activator domain that's called VP64. It's a very highly active protein domain that can activate a lot of transcription. And that's also fused to blue fluorescent protein so they can follow the dCas9. All right. So what they do then is they transfected the cells with these. So, all right. So what they're going to do is they're going to put the dCas9 with the localization sequence, with the blue fluorescent protein, with the activation domain, with the sgRNA, and measure the GFP fluorescence from the reporter gene here. And how does the data look like? Now they're putting the data in a different way. Instead of plotting the fluorescence signal from GFP and counting cells, what they're doing is here they're just doing a population assay. What they're going to do is they're going to make a chart where on the y-axis you're going to see the fluorescence from the GFP. So the higher the fluorescence, the more GFP expression, right? So this is basically measuring transcription. Higher transcription, higher fluorescence. And what they're plotting here is their different conditions. So in their different conditions, in one condition, what they did is they transfected cells with the sgRNA that is targeting these sgRNA target sites upstream of the GFP reporter, but they didn't put any of the dCas9 fused to the activation domain. And what you can imagine is that because you're not putting any activation domain close to the gene, you would see not much expression. So that's going to show you your baseline, right? And that's the baseline expression right here. Next, what they're doing is they're doing the opposite. They're putting transfecting the dCas9 that's fused to the transcription activation domain, but without the sgRNA, so that the Cas9 is not going to be able to bind to these sites, right? And so that's, again, measuring just the basal expression, and that's not very much either. Now, when you transfect the cells with both the sgRNA and the dCas9 fused to the activation domain, now this is going to be able to bind to these sites and activate transcription, and that's exactly what you see. You achieve 25-fold more GFP expression when you do that, right? So then you can modulate the activity, the expression of the gene by targeting the dCas9 fused to the activation domain. Is that clear to everyone? All right. What else can you do? You can do a whole bunch of stuff. This is another example of what you can do. What you can do is you can make an sgRNA or a collection of sgRNAs that are going to bind to a certain genomic site and bring the dCas9. And what you can do is you can fuse dCas9 to GFP, and whenever the dCas9 binds to these genomic regions with the RNA, and dCas9 is a remarkable enzyme in that its binding is very, very tight. So it has a very high affinity to where it binds. And it just like binds there and just pretty much gets stuck in there, okay? So once it binds to its target sites, because there will be lots of GFP signal from these sites, what you can do is you can live image the cells. And here, for example, DCAS9 was targeted to 3 loci, and you can see that these little 3 dots here inside live cells. And you can do live microscopy and see the movement of these genes and correlate it to various measurements that you want to make, right? All right. What else you can use for what else can you do with CRISPR? One of the most exciting technologies that involves CRISPR is of course gene therapy, right? So you can do things like the CD4 cells, which can be done inside the cells, or you can actually do it in the organism itself. So in this experiment, what they've done is they said, okay, can we actually create CRISPR monkeys in which that we actually edit their genome, the zygotic genome, and basically alter their genome, right? So to do this, what they've done is they injected, like I said, anything that you can inject, you can do this experiment. So what they did is after the fertilization, they injected the DCAS9 that was complex with a couple of different sgRNAs that are targeting a couple of different genes to make mutations. And then they injected these embryos, and then they put the embryos into surrogate mothers, which then had babies. And they looked at the baby's genome to see if the editing was there or not. So for this experiment, what they did is they actually apparently targeted 3 genes simultaneously. They had to inject more than 180 single-celled monkey embryos. They were able to get 10 pregnancies out of these from 83 embryos that were implanted. One of which, at the time that I took this image, so I don't know what happened afterwards, but one of which actually led to the birth of a pair of twins with mutations in 2 of the targets, PPAR gamma and RAG1. And so you could generate these mutant monkeys. So anything you can do in the monkeys, you can actually do in the humans because we're evolutionarily related, right? So you can use the exact same techniques on human embryos. And so there was a group in China last year who tried this. But of course, you're already horrified that this can be done in a human because there are a lot of ethical concerns, right? The question is, should we do it? Can we do it? If we do it, what happens? If we don't do it, what happens? In other cases that maybe you'd want to do it, right? For medical reasons. So they said, okay, let's do the proof of principle. Can we actually do this, right? So they targeted only 1 gene in this human embryo. They set out to edit this HBB gene, which encodes the human beta-globin protein. Of course, unlike the monkey experiment, they actually did not want pregnancies resulting from this, and they did not want new babies coming out of this. So what they did is they used embryos that they knew were not going to be viable, okay? Because they contained extra chromosomes. Remember in the lecture in one of the previous lectures, I was telling you that a lot of human pregnancies result in embryos that have contained extra chromosomes, and these are typically lethal, right? Except the Down syndrome, which contained the chromosome 21. Most of these were lethal and caused a majority of the miscarriages, right? So what they could do is they could take these embryos and use them because they knew that they're not going to develop into humans. So what they did is they injected 86 embryos and watched them for 2 days. Of the 71 embryos that survived those 2 days, 54 were genetically tested, and 28 of them showed a successful cut. And they were trying to replace it with homologous recombination with some other sequence, right? Because if you want to do gene therapy of a damaged gene, that's actually what you want to do. You want to actually correct. And you want to do that through homologous recombination by providing a donor DNA. So when they did that, what they found is only a fraction of those actually contained the replacement DNA. So the process was not very efficient, right? But at the end, they were capable of getting embryos with the correct editing. So this is definitely something doable. So the Chinese researchers said that in their paper, if you want to do it in normal embryos, you need to be close to 100%, right? So this is a pretty low efficiency. So, and his team also found a surprising number of off-target mutations because the complex could act at other parts of the genome that they did not intend to edit. All right. What else can you do? You can do other things. You can even use the system in ecology, changing whole populations. So instead of injecting and changing single embryos, you can actually change a whole population of animals, and this is what's called the gene drive. So what you do is you you stimulate biased inheritance of particular genes. So that is going to alter the entire populations. So a technique for changing wild populations, such as you can alter mosquitoes to slow the spread of malaria or dengue fever or Zika or whatever you guys like these days, right? Or whatever the doom that's coming towards us, except the orange one. So what you can do is you can actually put a gene that is going to be able to cut, that is going to be able to make a cut, right? So this could be, this would be the endonuclease gene that is going to cut a certain site in the genome. So what you can do is you can create a mosquito that contains this gene. You can mate it to the wild type. Now, if the endonuclease gene is capable of cutting the wild type chromosome, and it produces a double strand break, sometimes that double strand break can be repaired by non-homologous end joining. And what you will have is you will have one copy of the chromosome containing your gene drive, and the other one is going to stay wild type or mutant due to small deletions or insertions. But if this double strand break is repaired by homologous recombination, which starts with recession of these ends, right? And if that recession results in homology outside these the inserted gene, what will happen is that homologous recombination can, not always, but can lead to gene conversion, which you guys learned in a homologous recombination lecture, and now you have two copies of that endonuclease gene. Now, when that mosquito with these two copies then mates with another one, then you can modify the genome of the second mate, etc., and the gene that you inserted will spread into the population. Okay, so that's why it's called the gene drive. So some of this, like any technology, right? Don't get scared. Like any technology, if you use it for good things, it'll be good. If you use it for bad things, it will be bad, right? There's nothing special about genome editing than anything else in the world. One of the things that is very exciting about this technology is that you can actually use it for a lot of things that people just, if you can dream of it, you can do it. So some of you guys may know that some of the pig organs are very compatible with humans, but one of the problems in using pig organs for anything, for tissue transplants or anything like that, is that the pigs actually have a lot of retroviruses that are still active. So they can jump from one place to another. And that's not good if it actually jumps into the human genome, because we know that transposons can cause a lot of mutations, right? So in this experiment, what they've done was that they created a transgenic pig where they used a Cas9 enzyme and targeted it to cut at retroviruses and completely eliminate, so knockout. And the way that you can do knockout is you can generate two cuts on both sides, right? And if that gets picked up by non-homologous end joining, the two sides can be joined, and you basically removed the the retroviral transposon, right? So it's kind of like very similar to how you do mouse knockouts. I mean, you can use a selectable marker to do this so that you can make sure that you get what you want. All right. So what they've done is they basically eliminated all copies of this certain active retrotransposon. So apparently there were 62 copies. So basically they created a pig line, right, a transgenic pig that would be safe to use for any human transplantation type of experiments in the future, right? All right. But what the most excitement comes from is the combination of this technology with stem cell technology, okay? And I'm going to probably talk about that in the very last lecture of the semester because it's just exciting. But the idea is this. So if you have a patient that you want to be able to treat through gene therapy, what you can do is you can isolate fibroblast or whatever other cell types from this patient, and then you can make induced pluripotent stem cells in vitro by expression of these 4 transcription factors we learned about, right? You can express them and you can reverse the course of the differentiation and you can make a pluripotent cell. So that's the induced pluripotent cell. And then what you can do is if you understand the processes of differentiation well enough, so if you've done enough basic research to understand how to push these into one direction or the other, what you can do is you can first correct their genome using genome editing and then push them into becoming the sort of cells that you want them to become, right? And if you can then transplant these back to the patient, then you would be putting in cells that is coming out of the patient itself, so you're not going to have any immune response to its own cell, right? But you actually corrected whatever mutation was causing the disease. Does this make sense? So you could take it from the patient, fix it, process it, and put it back into the patient. And this is actually what people have been really excited about, and there is some progress in the past 2 years or so in this kind of treatments. I know some of you guys want to be doctors, so it's really important you guys understand that knowing how differentiation works, knowing how molecular biology works, will allow you to come up with more innovative techniques to treat patients in the future, right? Okay, so that's pretty much it for today. Have a great Thanksgiving, and I'll see you guys in 2 weeks or so.